This is Dr. Momcilovic's Bio 182 Lecture 4.2 on classification and taxonomy. So our modern way of classifying and naming organisms is based on the work of a Swedish a Swedish theologian in the 1700s by the name of Carl Linnaeus. Linnaeus was organizing different organisms for his study of theology. So it was essentially look at all of these different organisms that our God created. There's all these different types of life, all these different living things on earth. Look how great God is. We now know that, or we don't quite use the same way of looking at it. We now use his system for classifying organisms to look at the different relationships for different species. However, his naming system was so precise and so useful that we still use that exact same system today. So his way of naming different organisms is called binomial nomenclature, which means that each organism's name has two different parts to it. Bi meaning two, nomial, so two names. So each type of organism can be referred to by a unique two-part name. It's very similar to us having a first and a last name, but in this case, our last name would be first and our first name would come second. So my name would not be Ashley Momcilovic. My name would be Momcilovic Ashley, meaning that there's a group, right, of Momciloviches. I am the Ashley of that Momcilovic group. And so in binomial nomenclature, we have, instead of first name, last name, we have genus and species, where genus is essentially the last name and species is like the first name for that specific organism. For any organism on the planet, it has a genus and a species name, anything that has been named by science. So examples would be Homo sapiens, which are modern humans, that's what you are, and Homo erectus, which is an extinct human that lived about 2 million years ago. They're both of the same genus, which is Homo. However, they're different species within that genus, so sapiens and erectus. Similar thing when we think about gray wolves and coyotes. Gray wolves are Canis lupus and coyotes are Canis latrans. So when you write these out, it's very, very important that you write the genus species name in the correct way. So Homo sapiens would always be capital H and then followed by lowercase OMO space and then lowercase all of sapiens. Same thing for Homo erectus. Capitalize the genus name and then everything else, including the species name, is in lowercase letters. And so this essentially shows us that anything in the same genus is very, very closely related. So everything in the genus Homo is closely related. Anything in the genus Canis is closely related and is going to be similar to things that are Canidae, right? Things that are similar to dogs. And so this is very important and very useful. And when you write it, make sure that you write it in the correct way. So not only is the name going to be the first part, the genus will be capitalized and everything else, including the species, will be in lowercase. When you type out a genus and species name, it will always be in italics if you're typing it. If you were handwriting it for some reason, you would just underline it. It's very hard to write in italics, obviously, so just underline it if for some reason you're writing it by hand. But just make sure that you're always using that correct form with the capitalized genus, lowercase everything else, and then in italics if you're typing, or underlined if it's by hand. So we use Linnaeus's binomial nomenclature system to name and classify all of the different types of life on the planet. Within life, there are three domains. There's bacteria, archaea, and eukaryota. Bacteria are the most ancient. They would have been the first group to evolve. The first type of life, we believe, was bacteria. That would have been followed by archaea. Archaea are often called extremophiles. We'll talk a little bit more in detail about what that means. But archaea are simple single-celled organisms that live in very extreme environments, like very salty areas like the Great Salt Lake or near hydrothermal vents, things like that. They have different types of metabolism that allow them to live in environments that would not be suitable for any other type of life. Bacteria and archaea together are sometimes called the prokaryotes because they have no membrane-bound organelles. And that differentiates them from the eukaryota, which are the multi-celled organisms such as animals and fungi and plants and all that good stuff. So those are the three domains of life: bacteria, archaea, and eukaryota. Within the eukaryotic domain, there are three eukaryotic kingdoms. In probably high school, you would have learned that there were 5. There's actually 3. We don't do 5 anymore. We don't count protists as their own thing. We count them a different way. So we are now going to only refer to the eukaryotic kingdoms as being the kingdom Plantae, the kingdom Fungi, and the kingdom Animalia. All life on the planet, all species, every single living thing on the planet has an evolutionary relationship of some kind with every other living thing on the planet. So for any two species, or if we're talking about genera or higher groups such as phyla, every single way of categorizing and looking at the organisms that are alive on the planet, they all have some type of an evolutionary relationship. That means that somewhere in the past several billion years, there is a point at which those two organisms share a common ancestor. And following that common ancestor, when they branched off from each other, there was a divergence and then independent histories that have led those different species or genera or phyla or whatever we're talking about to their current state. So when we're talking about like leopards and cats, you guys have all seen like the commercials where, you know, your cat has a wild animal inside or your dog has a wolf inside, all that kind of stuff. And it's true, your domesticated animals are related to and descended from wild animals. They have a common ancestor, and at one point that branched off and we domesticated things. It's not just that though. You have a relationship also to your dog or your cat or your bird or your guinea pig, whatever you have in your home. You also have a relationship to the plants in your yard, the trees, the bacteria, every single living thing on the planet, even if it's billions of years ago, has a common ancestor with each other. So when we're talking about categorizing different organisms, it's not just at the genus and species level. It's also at several other orders of magnitude. So the most broad and all-encompassing would be life. All things in life on Earth that are living fall into that category of life. Within that, we have those 3 domains, followed by kingdoms, and then phylums, class, orders, families, genus, and species. And so species would be the most specific and the most exclusive. Kingdom and domain are broader and more inclusive. And so we work from the broadest of life, domain, and kingdom, which encompass tons and tons of different millions and millions of organisms, and work our way down to the very specific species level. An easy way to remember this, and most people can remember life and domain are sort of the biggest and broadest, to remember kingdom, phylum, class, order, family, genus, species. You can remember King Philip came over for good spaghetti. There's a bunch of other different mnemonic devices to help you do that. but just sort of so that you can remember the order in which they go: kingdom, phylum, class, order, family, genus, species. So when we're looking at the relationships between these different species or genera or phyla or whatever it is, we create what's called a phylogeny, which is a hypothesis of the evolutionary relationships between groups of organisms. So, all right, so you're going, okay, Dr. Momcilovic, it's a hypothesis. That sounds like we're guessing on the relationships. Remember that a hypothesis is something that we have some evidence for, right? We have a reason that we think that this is the way that these organisms are related to one another. And so a phylogenetic tree is constructed with the best current scientific information, and that includes the fossil evidence we've been talking about. It includes behaviors, it includes molecular evidence like DNA and RNA, all of those things, and all of that data is all constantly being reanalyzed, and we're looking at the relationships and reorganizing these phylogenetic trees and these different relationships to try to get the most current and comprehensive and best hypothesis, right? The best phylogenetic tree that we possibly can based on the data that we have. So if you're ever asked to look for a phylogenetic tree, you want to find the most absolute current phylogenetic tree for whatever it is that you're interested in, because that data can change so quickly that from maybe 2 or 3 years ago, or even last year, there may be a new tree based on new genetic information. And so you always want to make sure that you're looking at the most current version of the hypothesis of the relationship between those species. Okay, so a phylogenetic tree is just a diagram that illustrates the hypothesis for the relationships between organisms. So say that we have organisms A, B, and C. If you look at this first phylogenetic tree, you can sort of see by looking at it that organisms B and C seem to be more closely related to each other. They're located closer on the tree. A very important thing to look at is the branch point. So their most recent common ancestor is that branch point where one half is green and one half is blue. And so that's their most recent common ancestor. B and C are both equally related to A. It doesn't matter because we could flip-flop B and C on that tree. Their location doesn't really make a difference. It's just the fact of how far away from A they are. So B and C have the same common ancestor with A. And so A, B, and C are related more closely to each other, and they're equally related to A. So sometimes we have phylogenetic trees that are oriented this way with time on the vertical axis, where the most recent time, or the present day, is located at the top of the tree, and the further you go down the page, you're going further back in time. So you can see where these common ancestors are, and the length of these branches on the tree are telling you time. We can also do this where some other trees can be oriented on the horizontal axis, where you have present day species on the right, and we're going backwards in time as we go to the left. So it's the same thing. It means the exact same thing as in the vertical way, but they're just oriented differently. But in that case, we're saying that A and B are closely related. Their most common ancestor is shown. And then we're saying that A, B, C, D, and E all have a common ancestor that's further back in time. So all of those species are related to each other, but depending on how far away the branch point is that connects them determines how recent their common ancestor was, and therefore how related they are. One important feature of looking at the relationships between different species as well as phylogenetic trees are what are called ancestral traits. And these are features that are found in a common ancestor and are then inherited by subsequent lineages. So one example, which we talked about before, is the limbs in tetrapods. So humans, dogs, birds, and whales are all tetrapods because they have 4 limbs. And within that, Within the forelimbs, so the arm limbs essentially, right, of all tetrapods, they have the same bone structures. You have a humerus, which is the one that sort of goes from your shoulder to your elbow. Between your elbow and your wrist, you have a radius and an ulna. And then beyond your wrist, you have your phalanges, which are your digits. All tetrapods have those same bones in common. They're just different lengths and shaped a little bit differently because of Hox genes. And Hox genes determine how long they're developing and therefore how long they get. So when we think about like a whale, you don't think of a whale as being a tetrapod. You don't think of them as being a four-legged animal. But whales are technically tetrapods. And if you think about their flippers, they have very short sort of arms, right, that their flippers are sticking off of, and then their flippers are full of those 5 digits. If we think about something like a dog, they have a little bit longer of an arm and then sort of shortened phalanges. And so if we look at this chart sort of to the right, we can see where that common tetrapod ancestor is, and everything that came after it has that same structure. And based on the Hox genes, we are able to, or they all have different ways that looks depending on the Hox genes and how long those genes are turned on for development. So starting around like about 380 to 360 million years ago is where we saw the first tetrapod, and everything that evolved past that branched off from that common ancestor has that same tetrapod arm. And so ancestral traits are going to be really important and something that you should be watching for, something that says that this common ancestor had this trait and everything after it also had those. So that's something you guys should definitely be watching out for. And as we go through some of these different groups, I would advise you to make flashcards with info on shared derived traits and things for each group. So a shared derived trait are features that evolved uniquely within a clade, and these features are evolutionary novelties within their group. So for example, on the following deuterostome phylogeny, the shared derived traits are marked in blue. So milk is a shared derived trait for all mammals. Chordates, so having a notochord is a shared derived trait for all chordates. And so you can see along the bottom in blue where we have vertebrates, we have things with 4 limbs, we have things that are amniotes, and anything that comes after that shares that same derived trait. And again, it's a shared derived trait is a feature that evolved within a clade, and it's a novelty within that group. And all members of that group have that same trait. When scientists are building phylogenies, they use a lot of different types of data, and we update those phylogenies when we get new data. So historically, one of the most commonly used types of data and information that we used to build a phylogeny was morphological data. So how things looked. Hundreds or so years ago, scientists wouldn't have had the same data and the same information that we have today to be able to build accurate phylogenies. So they based all of their assumptions on the relatedness of species on morphology. And so there's a couple issues with that. So sometimes we think certain things look the same and therefore they're related. And that would be something that we call convergent evolution, where 2 different species have a similar appearance because they have a similar environment that they live in, or a similar need that they need to meet in order to survive. So if we think about like bird wings and bat wings, birds and bats are both flying organisms, and so they both have wings. Their wings are structured internally very differently, And they have those wings for different reasons. Bats are mammals, and so wings developed sort of independently in bats. And then birds are technically reptiles, and so wings developed separately in birds. And so wings developed in two different groups, and they look the same because they are both flying animals, but they are not the same because they got their wings from a common ancestor. So that's what we call convergent evolution. A similar thing would be something like the fins of like a shark and the fins of a whale or a dolphin. A shark is a fish, a whale is a mammal. They both have the very similar looking fins because they have to move efficiently through the water and be able to steer. So they have sort of the same dorsal and pectoral types of fins. But they are that way because of convergent evolution, where their traits have converged on each other and look similar because they live in a similar aquatic environment, not because they got those shapes of their fins from a common ancestor. Another issue with morphology would be divergent evolution, which is where some organisms that are closely related look very different from each other, and it would be hard to determine that they are in fact close relatives. So a good example would be the finches on the Galapagos Islands. If you think back to that sort of radiation of them, they all look very different. They're different sizes in general. Their beaks are different sizes. They eat different things like that. And so it's very difficult to imagine that they all came from a common ancestor because they're so specified to their habitat, but they are in fact from a common ancestor. And so morphological data can be really useful, but it can also be a little bit misleading. So we don't generally base a phylogeny just on morphological data. We would base a phylogeny on morphology with some of these other things. So we could base it on development. A lot of warm-blooded animals a lot of mammals look very, very similar when they're a fetus, and then those differences don't start to appear until later. We can look at, to make our phylogeny, we can look at paleontology. So that would be those fossils that we talked about. And we can look at the differences and the similarities between organisms and things that we think were ancestors of that, and see the different characteristics and traits that are inherited. And so we can sort of look backwards and try to see the ancestors of modern day organisms. We can also use animal behaviors to try to create a phylogeny or inform our phylogeny behavior. Different things behave different ways, right? And so if they behave similarly, they're possibly closely related, things that burrow or things that fly, things like that. But again, it can be misleading in a similar way to morphology. And then the most recently developed and the most powerful tool that we have in creating a phylogeny would be molecular data, which is that DNA and RNA. And we are constantly able to test the genomes and sequence the genomes of different species, and we're constantly getting more and more information. So if you would have taken this class, 20 or 25 years ago, it would be very different when molecular evidence was hard to get, it was hard to sequence, it was expensive, it was time consuming, and technology has come such a long way that now molecular evidence is actually the most powerful tool that we have in determining relationships between organisms. But we still never base a phylogenetic tree just on molecular data. We also take into account these other things that we know can be similar and different between organisms, and try to build the best and most comprehensive tree based on all of these different types of data and evidence. So within a phylogenetic tree, there's a few different terms that we use to describe groups of organisms. So the first one would be monophyletic, or a monophyletic group. And this is where we have an ancestor and all of its dependents, or all of its descendants, excuse me. So in chart A, we have our ancestor B and every single species that evolved from ancestor B. So C, F, D, E, G, and H, they all are descendants of B. So it's an ancestor and everything that descended from it. If we said it was ancestor A that we're interested in, it would be A and every single other thing on this chart, because A is the ancestor and all of those other species, B through K, are all the descendants of ancestor A. So again, a monophyletic group is an ancestor and all of its descendants. There's two other categories that we can make when we're talking about phylogenetic trees, and they're quite a bit less informative. But one would be a polyphyletic group, which would be a couple of organisms, but not their common ancestor or their relatives. So in this case, we're looking at just species E and G, but we're not looking at their common ancestor, which would be species B or any of the other relatives in it. We're just interested in those two taxon. And so it's polyphyletic, meaning that it's just a couple of organisms and nobody else, not their common ancestor or any of the other relatives. The third type is called a paraphyletic group, and this is where we have an ancestor and some but not all of the descendants. So in this case, we have ancestor A, and then the group that branches off and makes I, J, and K, but not everybody else that's in that group. So we have an ancestor and some of the descendants, but not all of them. So part of the reason that a polyphyletic group is not informative is because we're looking at the relationship between two organisms, but not anybody else. So if we used to think about mammals and birds as being closely related because they're both warm-blooded vertebrates. We now know that birds are actually a type of reptile. And so thinking about birds and mammals as both groups being warm-blooded is misleading, and it brings us to wrong conclusions about the evolution of different animals. So birds are actually more closely related to the crocodilia, the lepidosaurs, and the testudines. So those would be like the lepidosaurs are lizards and snakes, and the testudines are turtles and tortoises, those kinds of things. And crocodilia, obviously crocodiles and alligators, things like that. So birds are descended from types of reptiles. So they're actually more closely related to crocodiles than they are to mammals. But if we just think about them as being warm-blooded, then we would assume that they are closely related, but in reality, they create a polyphyletic group. The other one, using the same example, would be paraphyletic. So if we're saying reptiles, we're saying, oh, it's reptiles, so it's, the crocodiles and the alligators and the snakes and the lizards and the turtles and the tortoises, it's all those guys, but we're leaving birds out. Then it becomes a paraphyletic group because we have an ancestor and some, but not all of its descendants. So we have an ancestral reptile ancestor and then some, but not all of its descendants. So birds are actually a descendant of a reptile. And so by excluding them, we're creating a paraphyletic group instead of a monophyletic group.